Our next major topic in the semester is evolution, and chapters 13, 14, and 15 will be looking at various aspects of evolution. And so in this presentation, just want to introduce the idea of evolution and some basics about the evolutionary process. And evolution is very much a central theme in biology. Whether you're studying species or crops or medicine, you really can't learn very much if you don't take into account evolution. Despite this fact, it certainly is a very misunderstood idea, whether we're looking at just biology or even in all of scientific. Evolution oftentimes brings up lots of emotions, lots of different ideas. And so today what I want to do is just talk a little bit about evolution in the strict biological sense of the word, what exactly we mean when we talk about this process of evolution. And as a biologist, we certainly think of evolution as a process, some sort of process of change. And our biological definition of evolution is the change in allele frequencies in a population over time. So if evolution is occurring, we would expect allele frequencies in my population to change from one generation to the next. So I just want to take a few minutes and break down our definition and look at some important aspects of our definition here. So our first idea, changes in allele frequencies in a population over time. So what exactly are we talking about when we talk about allele frequencies? And we spent a whole several chapters here talking about alleles, those alleles being alternate forms of genes, and we talked about Mendel and variation beyond Mendel and those aspects. But instead of individual alleles, whether you've got big T for tongue curling or little t for not curling, when we think about evolution, what we're really interested in are allele frequencies. How common are those different alleles within a particular population or a particular group? And one of the big changes we're going to make from here on out throughout the semester is we're really changing our scale. Up until now, we've really talked about the scale at the level of an individual organism. Each individual has two alleles for a particular gene. How do those different alleles maybe affect the phenotype, or maybe we'll do a Punnett square and see which two alleles you may pass on to your offspring. But we were looking at things really at the level of the individual. But now as we start thinking about evolution, we're going to take a step back and we're really going to think about groups of individuals. We're not really interested so much whether big T is going to cause your tongue to be able to curl or not. Instead, we're more interested in, well, how common is that big T in our group, and is it going to become more or less common as time goes by? So this is a really an important misconception that I want to make sure we're all straight on right away, is that we're not interested in individuals as much as we are the group dynamic when we think about evolution. And the idea of allele frequencies is very straightforward, and let's just look at a quick example, and I think sometimes it helps to see some numbers with these examples. So let's think of that widow's peak allele, and big W, remember, was widow's peak. That was dominant. Let's say in my group I've got 15 people that are homozygous for big W, and I've got 30 people that are heterozygous, big W, little w. And then I've got 55 people with a straight hairline. They would have two little w's as their alleles. And I just pick these numbers because it adds up to 100, and that always makes math a little bit easier. So instead of being interested in who has a widow's peak and who doesn't, or who might pass it on, in evolution now we're interested in our group dynamic and how common are these alleles. And so we can ask the question, how common is that widow's peak or big W allele? And with this kind of data with genotypes, we can just count. If we look at that first group, there's 15 people, but each of those people have 2 copies of the big W. So that's a total of 30 copies of the big W or widow's peak allele. In my next group, there's 30 individuals, but each person in this group only has 1 copy of the big W, and so that's another 30 big W's worth of genetic information. And then obviously in my last group there aren't any big W's, they're all little w's, so there'd be none. So in my group, I've got 60 big W alleles. Okay? Now we're interested in a frequency, which means it's sort of like figuring out your grade. You're going to take your points and divide it by the class total to get sort of your percent or your frequency. The same thing here. Now if I've got 100 people, remember, each person has 2 copies of each allele, which means in my group, I've got 200 total alleles. Okay? So 200 alleles, 60 of those 200 are big W, so my frequency of big W would be 60 out of 200, or 30%. Okay? And so again, we always take our count and we're divided by the total number of alleles. We can certainly do the same thing for little w. How common is that straight hairline or little w allele? Obviously, there's none in my first group. In the heterozygous group, I've got 30 people, and everybody has 1 little w, so that's 30 little w's. And then in my last group, I'm going to have a whole bunch. I've got 55 people, everybody's got 2 little w's, so that's 110. So in total, I've got 140 little w's out of, again, 200 alleles in my population, because again, every person in my population has 2 alleles. So my frequency of little w is 70%. So if I could put these alleles for this population in a hat, and I pulled out 10, I would expect on average 3 of those to be big W's and 7 of those 10 to be little w's. And now notice if I add those two things together, big W and little w's frequency, I come out to 1 or 100%. And that should make good sense because in this case, I've only got 2 alleles, a big W and a little w. So if it's not one, it has to be the other. And so we can always check our math on these kind of things and make sure they add up to 100%. Our next important aspect of our definition is the idea of population, changes in allele frequencies in a population over time. Now, population just refers to a group of the members of a species that live in a particular area at a particular time. And so organisms don't tend to live just randomly scattered out there in nature, but instead they tend to live in very distinct groups or populations where all the members of the species are going to survive. Now, most species are made up of many populations. Maybe the species occurs in 10 different places or 15 different places. If it was a really rare species, maybe it only occurs in 1 or 2 populations, but that would be the exception, not the rule. Now, when we think of a population, though, again, in our evolutionary sense, we're not interested in each individual in the population, but what I'm really interested in are the allele frequencies or the alleles in that population. And so we would call that a gene pool. The idea that each population has its own pool of genes or collection of all the different alleles that the individuals in the population are carrying around. And again, in evolution, that's what we're interested in. Are those pools of genes, and are those gene pools changing over time, or are they going to stay the same? Now, the concept or the idea of a population is absolutely critical when we start thinking about the process of evolution. So let's take a second and look at that as an example. Let's say I've got a species that lives in 3 distinct populations, and we'll just label them populations 1, 2, and 3. Let's say it's a species of salamander. Okay? So I've got regions where this population, where the species survives, and those populations are separated by regions where I don't find that salamander. And what determines where I find the species and where I don't find the species is basically the habitat or nature. Okay? Species are well adapted or well suited to live in particular areas. In the case of a salamander, maybe it lives in places that are shady and moist and cool. And so in population 1 there, I have a big valley with a lake in the middle. Or maybe in population 3, I've got a stream bed, and right along that stream and that valley, I see my salamander. Okay? But in areas where I don't find it, that habitat is very inhospitable. Maybe, for example, it's a really high sunny plateau, and so I don't find the salamander in between those different populations. Okay? So each population represents members of the same species occurring in different places. Now let's say, for example, in population 1 I labeled 2 salamanders, and let's just call them salamanders A and B. And in population 2 we have salamanders C and D, and 2 more, E and F, in population 3. Okay? And we were to look at individual salamander C, and that's the one we're really interested in. And we could ask the question then, looking at individual C, who is individual C most likely to mate with when it comes mating season for salamanders? Okay. And it's not a trick question. The answer here is obviously D, for a couple of reasons. Number 1, D is closer. So all else being equal, C is more likely to bump into D during mating season than it would be to bump into A or bump into F, for example. But not only that, but if C technically could mate with A, but it would just be very unlikely because not only would one of them have to cross a long distance for them to get bump into each other, but somebody's going to have to cross that inhospitable habitat where the salamander just doesn't do very well. So one of the reasons a population is so important to us is because populations represent that pool of genes where most of the mating is going to occur. Okay? And our take-home message here, thinking about individual C, is mating is much more likely to occur within populations than between populations. Let's let that sink in. Mating is much more likely within populations than between populations. Again, C and D are much more likely to mate with each other than C and A. Okay? Which means in population 2, every generation, those individuals are going to mate mostly with each other and pass on their alleles. And that's going to determine which alleles we see next generation. Okay? So as we think about evolution and allele frequencies, we're really interested in who survives long enough to become mature and who's mating, because that's going to tell us what alleles are going to be present next generation. And so we can think of each population as having its own separate pool of genes, and every generation that population is going to mix up its genes. Some are going to get passed on, and some are not. And so that's why we think of our population as our unit of change. Now, we've got C up there circled, so if we're thinking of individual C, can that individual C, that salamander, evolve? And the answer is no. Individual C has its genes, and its genes are not changing. And so this is another kind of common misconception with evolution. Individuals are not changing in evolution, and that's certainly not a prediction or not what evolution says. What evolution says is that populations are going to change. Populations are a unit. And so, for example, maybe in population 2, I've got 30% big W and 70% little w are my allele frequencies. If I come back in 50 years, and maybe all those salamanders are dead, but I look at their descendants in that population, who they pass their genes on to, and I see that big W frequency is now 50% and little w is 50%, then I would say that population has evolved because it's not the same as the original 30 and 70% that we looked at. Our final part of our definition then of evolution is time. Changes in allele frequencies in a population over time. And certainly time is an important aspect to how evolution works or our process of evolution. Now as humans, we measure time in absolute time. How many years have gone by, or decades, or centuries, or minutes. But when we think of organisms and evolution, really that's not a very accurate measure. A better way to measure the time aspect would be thinking about generations. Right? How many times has this species mixed up its DNA and passed on its genes, for example? Whether it's been 5 generations or 100 generations or 1,000 is more important than whether it's been 10 years or 50 years or 100 years. Okay? And so because of that, when we see these kinds of studies looking at population genetics, changes in allele frequencies in our population, typically we see biologists study organisms with really short generation time. For example, maybe I'll study some bacteria who can crank out a generation every half hour to an hour. Or maybe I'm going to study insects that might have 3 or 4 generations over the course of a summer. Or maybe I'll study annual plants that have a generation each growing season. Because in my relatively short lifetime as a human biologist, maybe I can study 50 or 100 generations to look for any change. Versus I could certainly study elephants if I want to look at population genetics, but I'm probably going to have to wait a long time to see a few generations go by, and I might not be around to see the results of the end of that kind of study. So if evolution does consist of changing allele frequencies in a population over time, the next question that kind of pops up is, well, what would cause allele frequencies to change? If allele frequencies are changing, there must be forces out there in nature that are causing that change. And we would say, or label anything that causes change to be a mechanism of evolution. And there are 4 main mechanisms that might cause these changes. And let me just list them here, and then we'll look at them in a little bit more detail. Mutations could cause evolution, a process called genetic drift, migration, and then finally natural selection. So we're just going to work through these 4 mechanisms today and look at, first of all, what is each mechanism, and then second of all, how they might cause change in my population. And we've already talked about mutations, so mutations is a good place to start here with our mechanisms. Now we've talked about mutations as mistakes in the DNA copying. We said mutations are very rare, but they do occur, and we could get a frame shift or a point or a nonsense or silent. We talked about lots of different mutations. But again, when we talked about those mutations, we talked about them at the level of an individual. How does that mutation affect my proteins, or maybe how does it affect my offspring if I pass on that mutation? Now we're much more interested in our group dynamic. How are these mutations going to affect the population or the group as a whole? So let's look at an example here. Here's our original numbers from our population allele frequencies before. We said we had 30% big W's and 70% little w's. Now let's assume this population is going to reproduce and make the next generation, and just for simplicity's sake, we'll keep everything exactly the same. So we're going to still have 100 individuals next generation. We're still going to see our alleles being passed on. The only thing is, though, we're going to have one mutation. When one of those big W's is getting copied during DNA replication, instead of getting copied correctly, we have a mistake. And it gets copied into something else. Let's call that mutation W prime. Okay? So just one out of those 60 big W's gets copied incorrectly. So now I have this new W prime. Maybe it was a point mutation. Okay? So if I compare now, how has that changed my population? Well, I'm not going to have 60 big W's anymore because one didn't get copied correctly. So now I'm going to have 59 big W's. Okay? Still 200 total in my population, so my frequency of big W's changed. Now it's a really minor change. It went from 30% to 29.5, but technically still a change. If we look at the frequency of little w's, I've still got 140 out of 200, so I really haven't changed my little w frequency at all. Okay? But I've got one more allele now to keep track of. I've got this new thing, this W prime. Now it's really rare. There's only one copy of that mistake, but we still have to keep track of it. So 1 out of 200 would be my W prime. And again, notice if I add up all my allele frequencies there on the right, I'm going to get 100%. So looking at our effects of mutation on our big picture of evolution, we can see while technically it caused some change in the big and little W frequencies, it really was a really small, almost negligible change. It changed big W from 30 to 29.5, but extremely minor. But mutations do play a big role in evolution because they generate new alleles, that new W prime. In fact, the only way new alleles arise is through mutation. We get new combinations of our A, T, G's, and C's. And so very, very important when we think of evolution is the generation of new alleles, the process of mutation. Now, chances are this mutation might be detrimental. And if it is, the individual that has it might not survive, and that mutation may just disappear from the population. But if that mutation was a benefit, maybe that individual survives and passes it on, and if I came back to my population in, let's say, 50 generations, maybe half the population has that W prime. Well, in that case, I wouldn't really think of it as a mutation anymore. I would just call it an allele. And so all of our alleles started as mutations at some point, and some have disappeared, while others have become more common over time. Our next mechanism that might cause change is called genetic drift. And genetic drift refers to the idea that sometimes allele frequencies may change just due to chance or random events. Remember that when we think of genetics, we had our Punnett squares, and our Punnett squares did a lot of math for us. That it's all about probability. If I'm big W, little w, I expect half my kids to get a big W and half to get a little w, but I certainly wouldn't be shocked if I passed on 3 big W's in a row, for example, and never passed on little w. And so because genetics is based on probability, I expect this to happen, but I don't always necessarily see that happening on a small sample size. Chance or random events may have a big impact on genetic drift, on the allele frequencies in a population, causing genetic drift. And we said when we thought about our experimental design or when we thought about genetics, if I've got a small sample, that's going to be affected in a big way by random or chance events. If I flip a coin 1,000 times, I'm going to expect to see 500 heads and 500 tails. If I flip it 5 times, I might see 5 heads. Okay? So small samples are going to be affected by probability much more often than large samples are. And so if we think of genetic drift, then changes in allele frequencies due to chance, this may play a big role in populations that tend to live in small groups. And so if I have a small population size, random events may have a big impact on next generation's allele frequencies. And so genetic drift might be an important player when we think about mechanisms of evolution. So if we take a look again and use some numbers just as an example, let's say we've got 2 populations, and we'll make these populations exactly the same, except we'll make one small and one large. So here's population 1, and we'll make it really small. We'll have 5 individuals. And let's say 4 of them happen to be big W, and 1 is big W, little w. Well, if I figure out my allele frequencies, I've got 10 alleles, 9 of them are big W's, and only 1 is little w. So my allele frequencies are going to be 90% big W, 10% little w. Okay? Again, let's look at a second population, and we'll just add some zeros. That way we'll keep all of our genetics the same. It's just going to be a bigger group. And so in this case, 900 of my 1,000 alleles are going to be big W, 100 out of 1,000 little w. So again, mathematically, 90% and 10%. They're exactly the same. Okay? But let's say we have a random event occur to each population. Let's say one individual gets killed off randomly when it's a juvenile, before it has a chance to reproduce. Let's say a flash flood happens to wash through and it drowns one of these salamanders before it's matured and can pass on its genes. Okay? And let's say that salamander happens to be one that's big W, little w. Okay? Now obviously that's going to have a huge impact on population 1 because there was only one big W, little w to start with. And if we wipe it out, now my population only has big W's left. And so that one incident, that one individual being killed off dramatically changes my allele frequencies. Now they're 100% big W and 0 little w. And we would say that this population is fixed because now everybody's exactly the same. Everybody has big W's. And once that little w is lost, it's not like we can just get it back really easily. Right? We would have to have a perfect mutation for big W to mutate into little w, or perhaps maybe if little w exists in a neighboring population if it moved in. But a lot of times, once an allele is lost, it's lost forever. Okay? Now, one big W, little w being killed is going to have a pretty minor effect on population 2. There's still 99 of them left. And in fact, if we look at the math, technically it causes change, our frequencies go from 90 and 10 to 90.1 and 9.9%. But again, that's a really negligible change. And by the time I round off my fractions, really it hasn't caused much change at all. And so my small population is dramatically impacted by one random event. My large population, not so important. And there are a couple of special cases of genetic drift when populations are particularly small, even for a short period of time. And one is called the founder effect. And the idea here is we have a new population that's founded or started from just a few individuals. And so that founder effect is going to have to do with the genes from those few individuals that happen to found that population. That's a common thing we see with islands and mainlands, for example. We may see millions of individuals in a large continental mainland population, and a few of them will start a new population on an island. And those island alleles then in a population may be really different than the mainland, just depending on which few individuals happen to make it to the island. In humans, we see that with, especially here in the United States, with different religious isolates. Groups that came over in the early 1800s, for example, from Europe, often were these religious groups trying to escape religious persecution. In the eastern U.S., like Pennsylvania, we have groups like the Dunkers, which originally came from Germany. Here in the Midwest, we're probably more familiar with the Amish. Okay, and these are groups that were founded by maybe 50 to a couple hundred individuals, and which people started these populations were pretty random. Which people happened to get on the boat at the same time and start these populations. And even today, for example, with the Dunkers, while there may be millions of people living around the Dunkers still remains a relatively small, isolated populations because Dunkers are going to marry and reproduce with other Dunkers. And if you don't, you're not going to be a Dunker for long. You're going to be removed from the population. And so we still have these relatively small, isolated populations. And if we take a look at an example, Dunkers live in the U.S., they're descended from Europeans, but if we look at their blood types, which we know is inherited, it's different from both groups. The type A is much more common in Dunkers than either United States or European populations, and any of the B alleles are extremely rare. And this is simply due to chance alone, or this founder effect, that more individuals that got on the boat back in the early 1800s happened to be type A, and so we have a lot more A alleles as a result than what we would normally expect to see. Another special case of genetic drift is what's called the bottleneck effect. And this has to do with a population that if we looked at it today, and these northern elephant seals that we see in the picture is our classic example, today they number in the 30 to 40,000 range. And certainly by looking at pictures like the one we see here, they don't look particularly small population size. It looks like there's a lot of them around. And so we might look at the beginning and think, well, genetic drift, not important. Big populations here. But if the population has been small in the recent past, that may impact the alleles that we're seeing today. And if we take a look at these northern elephant seals, as we can see, they're relatively common. They spend a lot of time sunning on land and then spend time fishing in the ocean. And when people first started to arrive, settlers in particular in California, these things were very plentiful. We don't have exact counts, but they certainly numbered in the tens of thousands. And as you might guess, pretty easy to hunt. A good source of meat. Also, their blubber and fat was used for oil, for like lamps and so forth. And so they were hunted very easily. And by the late 1800s, there were very few known left. In fact, only 20 documented elephant seals existed in nature at that time. And so devastated by hunting. And in fact, this was one of the first species that was protected in the United States from hunting that almost went extinct. And so the population has recovered, and if we look at the bottleneck effect here with these elephant seals, today, again, they number in the tens of thousands. So we maybe think genetic drift not important. But remember, it's not absolute time. It's generations. And so while in absolute time this happened 120 years ago, the reality is for northern elephant seals, that was only about maybe 10 or 15 generations ago. And when we look genetically at these elephant seals, they're all very similar to each other. And the reason is they're all relatives. Which 20 happened to survive in 1890? That's a pretty random thing. 20 that happened to live in a place where no one had a gun or a harpoon. And those 20 then have passed their genes on to the 30,000 plus that we see today. And so that's the idea of this bottleneck effect. The small number in the past can have a big impact on today's genes. Next up then is the idea of migration as our mechanism of evolution. And migration, we're not talking seasonal migration, ducks flying south for the winter, for example. But in evolutionary biology, when we talk about migration, we're talking about individuals moving from one population to another. Okay? So if I've got two populations, we said that they're separated by inhospitable habitat, and most individuals are going to stay in a population and mate. But on occasion, we may have an individual get up and physically move from one population to another. Okay? And when that individual moves, the reason that this is important as we start thinking about evolution is individuals are going to take their alleles or their genes with them. And so in this case, the migration of this polar bear is going to impact both populations, because population 1 is going to lose some alleles, population 2 is going to gain a few alleles. Okay? Now, if only one, in this case polar bear, is migrating from one population to another, migration really isn't going to have a big impact on our allele frequencies. But if the species as a whole tends to be more mobile and tends to move around a lot, migration may play a big role in its evolutionary history. And as we think about evolution, remember, we're not really interested in the individual as much as those individuals' genes. And so we have a term here called gene flow that refers to the movement of alleles moving from one population to another. Remember, we said each population has its gene pool, so gene flow would be genes or alleles moving from one pool to another. Now, obviously, gene flow is caused by migration. As those individuals move, they bring their alleles with them. And again, gene flow may be important or not important to a particular species' evolution. It really depends on each particular species and how much they tend to migrate or move around. And we can look at a nice example of migration if we look at a type of baboon. And in particular, if we look at a species of baboon called the Hamadryas baboon. And these baboons are native to eastern Africa, the very eastern kind of tip of Africa, and also parts of the Arabian Peninsula as well. And like most baboons, they have very social clans where they have big social groups and extended families tend to live together. But when females mature, instead of staying and reproducing in their own clan, they tend to migrate or move to a new population. Now, this is actually pretty common among several species of mammals, and it's thought that this is a way to avoid inbreeding. Because if you stay and mate where you were born, you may be mating with relatives. But if you move to a completely new place, you're going to be mating with an individual that's not going to be related to you. And so, for example, here we can see some of these Hamadryas baboons, and again, their distribution, very eastern part of Africa and parts of the Arabian continent. Now, migrating is certainly a big deal to species because they have to cross inhospitable habitat, so that certainly carries a risk of predation or not surviving that inhospitable habitat. But many species like this baboon tend to do that as a way, again, to avoid that inbreeding depression. And so in the case of the Hamadryas baboons, if we want to study their evolution, we certainly have to take into account migration and where are these females moving, which population are they leaving, which population are they arriving at. Our last mechanism of change or evolution is natural selection. And certainly my guess is if I were to ask you about evolution, that most people think of natural selection first. Okay? And certainly this is where Darwin fits in. And he's one of those handful of people that you really should know their contribution to. And Charles Darwin wrote Origins of Species by means of natural selection way back in 1859. I do want to point out, again, some common misconceptions here. Darwin absolutely did not come up with the idea of evolution. Now, evolution as a concept back in 1859 when Darwin was doing his work, that was way before we knew anything about alleles or genes or a lot of the kind of concepts that we have today. But the concept of evolution in 1859 was that species are changing over time. And it turns out this concept of species change was around long before Darwin. In fact, it wasn't even biologists who came up with this idea of change, it was geologists. Because geologists in the late to middle 1700s were looking at fossils for the first time. And as geologists dug into deeper and deeper rocks, they realized the older fossils started to look more and more different than species we see today. And that led them to the idea that species must be changing over time, and that's why they don't look the same today as they did in ancient time and these kind of fossil records. So Darwin is not famous because he came up with the idea of evolution. That's absolutely a misconception. That idea was around a long time. The reason Darwin's important in our biological history is he was the first person to propose a mechanism of what would cause species to change over time. And these three mechanisms we've talked about already, mutation, drift, and migration, those are sometimes referred to as the new synthesis. And these ideas are all within the last century. As we learn more about DNA, we know that these mechanisms can cause change. Okay? But Darwin proposed the first mechanism, natural selection, long before these other mechanisms. In addition, these mechanisms that we've talked about, while they can cause change, they don't count for adaptations. And one of the real stunning things and amazing things about life as we look at different species on our planet is how many adaptations they are, meaning how well suited species are to live in particular areas out there. And when Darwin was explaining natural selection, he was trying to propose a mechanism that could explain how did species get adaptations? How did they get these specialized characteristics that let them survive and allow them to reproduce in their environments? And so I want to take a minute and look at this idea of adaptations, and then we'll move more into Darwin and his ideas about natural selection. So an adaptation is any trait that increases the chance of an individual to survive and or reproduce in its environment. And remember, these are the two important factors: who survives to become mature and who reproduces. That's going to determine our allele frequencies next generation. So as we think about evolution, those are the two important issues. And so an adaptation could be a physical trait or a behavioral trait that increase an organism's chance to survive where it lives. And a good example of that is this odd-looking little bird called the blue-footed booby. Now these blue-footed boobies, as you can see from the pictures, they live in coastal waters. They make their living fishing. And the picture on the right is sort of a time lapse of the same bird. And they go up in the air, they come zooming down toward the water. Right at the last second, they'll pull their wings back and they'll go diving deep to catch fish. Then they'll swim back up, they'll eat their fish on some rocky bank, and in the picture on the left, they have to dry out. And so he kind of has that odd-looking shape to dry his wings out, and then once he's dry, he'll fly and do the whole thing all over again. Okay? So if we look at this bird, it has several adaptations to its environment, despite its kind of odd appearance. First of all, we can really notice its bright blue webbed feet. Now the blue color has to do with mating, and we'll talk about those issues at a later date, but the webbing allows the bird to swim. Now they don't really need them to swim down very much because they can go pretty deep from the speed they get from flying, but they do need to swim back up to the surface to be able to breathe again. They also have specialized little flaps that cover up their nostrils. Most birds' nostrils are just open, and if they dove into the water, water would rush in and they drown. But these blue-footed boobies have little flaps that stick up that allow the bird to breathe, but then when it goes into the water, the rushing water acts to push those flaps and plug up the nostrils. Also their shape. And while the bird looks kind of odd while it's drying there in the picture on the left, right before it enters the water on the picture on the right, we can see they take this very aerodynamic, streamlined, muscular, kind of long and lean shape, and that allows them to dive very deep, very quickly to get their food. Okay? So even just looking at an organism very quickly, we can see several adaptations that allow it to survive in its environment. And certainly Darwin was asking the question, how did blue-footed boobies get these adaptations? And he explained through natural selection a mechanism that accounts for that change. And again, it's amazing to look at the natural world and see all the different adaptations. The Galapagos tortoise we see here with its giant shell. And there's a few of those around in the area. If you go to Grant's Farm, they've got a couple of these. Certainly the zoo has a couple of these giant tortoises. In the Galapagos, it's an odd place. These were the largest organism. There were no large mammals until people brought livestock there. And so that big, thick shell certainly provides a lot of protection. Speed certainly is a huge adaptation. Now that involves lots of traits, whether it's the shape of your bones or your muscles and so on. But the faster you are, the better chance you are to get food, the better chance you have to survive and reproduce. Okay? But probably the most common adaptation we see in nature is camouflage. And if we look at these mantids, and mantids are from South America, and they're basically insects that tend to eat other insects. Now the picture on the right, we can see this mantid pretty well. It's not only the same color of the leaves, it's the same general shape of the leaves. It even has a vein pattern that looks like the leaves, and it even looks wet. And it's really not wet. It produces this waxy chemical that makes it look like dew, so again, it blends in very easy. And while it's easy to see this in a close-up picture, chances are if we were walking through a Central American rainforest, we'd probably walk past 100 of these before we actually saw one. Okay? On the left, that one's even more camouflaged. And the only way to actually tell where it is if you look in the left side of the picture, you can see its antenna just sticking out a little bit. So that's actually its head pointing down. Its legs, it develops these little petal-shaped things on its legs that look very much like this flower, and it actually has a false eye spot on the end of its abdomen, so that if a bird, for example, sees it and takes a bite, maybe it loses part of its tail, but it doesn't kill the whole thing. And so this is a specialized mantid that lives only on this one species, and it'll hang out there all day, and then if a pollinator like a fly or a bee comes by, it'll make a meal out of that. So again, truly remarkable, some of these adaptations, as Darwin tried to figure out how did these adaptations arise. So as we sum up some of our basics of our evolution introduction. Again, evolution refers to changes in allele frequencies in a population over time. So our population is our unit of change. Individuals do not evolve, but the group is changing from one generation to the next. When we're talking about our different mechanisms of evolution, we talked about mutation, small changes in existing alleles, but important because it generates new alleles. Genetic drift, where we have our examples of the founder effect and bottleneck effect, important if we've got small populations, those random changes. Migration or gene flow, as individuals move from one population to another. And then natural selection. And we talked about the idea that this was Darwin's mechanism, the first mechanism proposed of all these, and that it's going to lead to adaptations. And so next time we'll start up with the idea of natural selection and our final mechanism there of evolution.